movement in and out of cell. So there are three possible processes that are involved in the movement of substances in and out of cell. In the last class, we studied about the cell membrane, and I told you guys the cell membrane is responsible for controlling the exchange of substances across the membrane, right? So when we are talking about this exchange of substances, there are three major processes involved in it. We have diffusion, we have osmosis, and we have active transport. So what are these three processes exactly? Let's start with diffusion, which is the most basic one and the easiest one to understand. Now, when let's go ahead and understand first a couple of concepts. Imagine that you guys imagine that you have a glass, and in that glass you have water, and inside the water you will have your water molecules, right? Now what happens, you went ahead and you dropped drop of color, a drop of purple color into the glass. What is going to happen? After a while, you will see that the purple color has spread all around and gotten mixed with the water. This is known as diffusion. The molecule or the purple dye diffused and it got mixed in with the water. What is actually happening over here? This is where you added the drop of purple color. At this particular point, the concentration of the purple color is high, right? Because you have a lot of purple color in this particular area. You have just put a single drop. So this particular drop has a really high concentration of that purple color. It has a really high amount of that purple color molecules. The rest of the water has low concentration of purple molecules. It has absolutely no concentration of purple molecules. At one particular point where you have put the drop of purple color, you have a lot of purple molecules. In the rest of the water, you have low concentration of purple molecules. What does this do? This creates a potential gradient or a concentration gradient. Let me explain this a bit better. This is the concentration, or this is the point, for example, where you have a really high number of purple molecules. This is where your water is. Your water has really low concentration or really low number of molecules. Between the two of them, between the point that has high concentration and between the point that is low concentration, there is a concentration gradient. The difference in concentration, the difference in high concentration and low concentration creates a concentration gradient or a potential gradient. Just like, for example, you're standing at the top of a hill. The top of the hill is high concentration, the bottom of the hill is low concentration. So just like what is going to happen when you are walking down the hill, you don't have to use energy, right? You don't have to do anything at all. The gravity will automatically help you walk down quickly and easily. So that is because of the gradient. That is because of the slope. The slope is helping you walk down without using much energy. That is what happens to molecules as well. So you have one point that has high concentration and one point that is low concentration. This difference in concentration creates a gradient, concentration gradient or potential gradient. And in diffusion, what happens? This difference in concentration allows the molecules to move from high concentration to low concentration. The molecules will go from high concentration to low concentration. So molecules of the purple dye that were at high concentration, they will start to spread through the water. Why? Because they are moving to the area of low concentration. And this is what diffusion actually is. Diffusion is the movement of molecules or ions or particles from high concentration to low concentration down the concentration gradient. Notice my terminology. I'm saying down the concentration gradient because you're literally going down. You're literally going down the slope. You're going down the hill. And why does this happen? Because the molecules are moving randomly. We know that molecules are always moving randomly. So because of the random movement of molecules, they will end up going from high concentration to low concentration, down the concentration gradient. And why is this happening? This is happening because of the result of their random movement. Is this making sense?
Yes.
Arisha, is this making sense?
Yes, miss.
So this is what your diffusion actually is. So when you're talking about your cell membrane, your small molecules like oxygen, carbon dioxide, they can simply diffuse in and out of the cell. They don't need any other specific process. They can simply diffuse in and they can simply diffuse out. So normally what will happen, that carbon dioxide diffuses out of the cell, it goes into the blood so that it can be carried to the lungs and it can be removed from the lungs. And oxygen generally comes into the cell so that it can be used for different biological processes. So that is what diffusion actually is. Now, moving on to our, 1 second, sorry. Right. So when we are talking about diffusion, let's talk about diffusion in relation to our body, right? What is the importance of diffusion in our body? So one thing I've already gone ahead and told you about is oxygen. Okay. So when you are talking about it, when you're talking about it, we know perfectly well that your oxygen is going to diffuse into the cell and it's going to diffuse out of the cell. Now, when you talk about a small organism like an amoeba, right? An amoeba is what? It's a single cell organism. It's literally one single cell. So in small organisms, you don't need to worry about having lungs or having some kind of breathing system because they're so small that they can directly absorb the oxygen or the oxygen can directly diffuse in from the surroundings. For example, in plants, right? The plants, the carbon dioxide can directly diffuse into the leaf. That's it. So oxygen in small organisms, usually gases can directly diffuse into the body. In large organisms, for example, like humans and animals, so our body is so large that we basically we need to have a respiratory system. So in our body, we have our lungs, and the oxygen will diffuse out of the lungs into the blood, into the red blood cells, and from the red blood cells it will be carried to the different parts of the body, different cells of the body, where the oxygen will diffuse out and go to each individual cell. Now that is one way that diffusion is taking part in our body. Now when you are talking about it, let's talk about, for example, carbon dioxide. We know carbon dioxide is produced by the cells as a waste product. And once the carbon dioxide is made, the carbon dioxide diffuses out of the cell into the blood. The blood carries the carbon dioxide to the lungs, and then the carbon dioxide diffuses into the lungs, and then we breathe it out. By the way, oxygen is transported in your red blood cells. Carbon dioxide is transported in the blood, in the liquid portion of the blood. If you've done animal transport, you will know that liquid portion of the blood is known as plasma. So carbon dioxide is carried in the plasma, oxygen is carried in the red blood cells. Then let's talk about other types of gases. For example, nitrogen. When you're talking about nitrogen, when you're breathing in air, nitrogen will also enter into our body. Nitrogen will also be coming into the body, but it will also enter our body by diffusion. We will breathe in the air, and then the nitrogen will diffuse from the lungs into the blood. But nitrogen is generally non-reactive. So even though it enters in our blood, our body doesn't really use it. So it remains as it is, and it is simply removed from the body. That's it. But there is a certain situation or a certain condition which is known as decompression sickness. Does anybody here know what is decompression sickness? Does anybody have any idea? Okay, all right. So generally what I told you is that nitrogen gas is generally, nitrogen gas generally is not utilized by our body. It is present in the air, but we breathe it in and we breathe it out exactly as it is. We don't use it, we don't do anything with it. We breathe it in and we breathe it out. But there is a certain condition known as decompression sickness. Let me tell you what it is. So, when you have deep sea divers, those people, scuba divers who really go deep into the sea, so in that situation, what happens is that when they go deep into the sea, they have a lot of pressure on their body. They have a lot of water pressure and air pressure on that body. So that pressure makes that nitrogen dissolve in the blood. So normally what would happen, that nitrogen would simply, be removed from the body. That's it. But when you're under that huge high pressure, you're deep under the sea, that nitrogen dissolves in your blood. Now, that is fine. The nitrogen is dissolved in the blood. Everything is fine. The problem occurs when the diver comes back to the surface. When the diver comes back to the surface, the air pressure changes. It goes from being really high to being low. Now, when the air pressure comes back to normal, the dissolved nitrogen turns into bubbles. So you literally have bubbles of nitrogen in the blood. And then those bubbles of nitrogen can block your blood vessels and lead to death of the diver. This is known as decompression sickness. So nowadays, what they do is that nowadays what they do is that they make sure that they have these special chambers which are known as decompression chambers. This is not for you don't need to know about the chambers. I'm just giving you an idea. You need to know what is decompression sickness. So they have these special chambers, and basically what the chambers do is that they bring the air pressure up slowly, so nitrogen has time to slowly turn into bubbles and slowly get out of the body, instead of the instantaneous change. So instant change is bad. The instant change turns it into bubbles, and also those bubbles get trapped in the joints and then they cause severe pain as well. So that is another example of where diffusion is important in our body. Making sense? Does anybody want to ask something? All good, Monami? Arisha, all good?
Yes, miss.
Okay. Now we need to discuss about the factors that are related to diffusion. What are the factors that affect rate of diffusion? And this is super duper important, right? You need to know the factors affecting rate of diffusion. The factors that are affecting the rate of diffusion, one of them is surface area, right? Bigger the surface area, the faster the rate of diffusion. Remember that. Bigger the surface area, the faster the rate of diffusion. And where have we studied this? If we have done the topic of transport in animals, we have studied this already in the form of microvilli. In the intestine, we have villi and we have microvilli. What was the function of microvilli? To increase the surface area. So maximum amount of food can be absorbed from the intestine. So greater the surface area, the faster the rate of diffusion. Then what other factors do we have? We have concentration. gradient. The higher the concentration gradient, the faster the rate of diffusion. How do we explain this particular point? Imagine that we're standing at the top of a hill. One hill is very steep, it's really high. The other one is a bit flat. So when you are running down the steep hill, the sharp hill, if you're running that down, it will be easier to get down because it is steeper as compared to a flat hill. A flat hill will have less difference in the gradient, or it will have less of a slope, it will have less of a gradient. So it will be a bit harder to get down that slope. That the same logic applies over here. The greater the difference in the concentration, one place has really high concentration, one place has really low concentration, that will allow quick diffusion. If at the same time one place and the other place have almost the same concentration, then the diffusion will be really slow. So that is the second factor that controls the rate of diffusion. Then we are going to talk about our something that is known as we're talking about distance. Okay, so think of it this way, that smaller the distance, the faster the diffusion. An example of this would be your cell membrane. Imagine if your cell membrane was like really thick, right? Just think of the diffusion that is taking place. That diffusion would be so much slower because the membrane is so thick, you have to cover such a great distance to come to the other side. As opposed to a really thin membrane. In a really thin membrane, what is going to happen? The thin membrane allows fast diffusion because the molecule has to only travel a small distance before it can cross to the other side. So smaller the distance the molecule has to travel, the faster the diffusion. Then it also depends upon the size of the particles, right? Think of it this way, that smaller the particle or the less mass it has, the smaller or less size or less weight it has, the easier it will be able to move. It is a simple example that you might have studied in physics. You have a small car and a huge van. You will have to apply a lot of force to the huge van to make it move. Same logic applies over here. You have a small, the smaller the size of the particle, the faster the rate of diffusion, the faster it is going to be able to move from one point to the other, and the diffusion will take place quickly. So these are some of the factors that are going to be important for your diffusion. And when we talk about diffusion, diffusion is going to depend upon what? Diffusion depends upon diffusion is basically a passive process. Passive process meaning that we don't need any energy for it. It will simply happen because of the concentration gradient. Anybody wants to ask anything so far? Does anybody have any confusion? Another factor that it depends upon is temperature. Higher the temperature, the faster is your rate of diffusion is going to be, because higher the temperature, the more energy the molecules are going to have, and the more energy molecules are going to have, the faster they're going to move, and diffusion will take place fast. So these are all the factors that are related to diffusion. We are all good, right? Nobody wants to ask anything?
Yes.
Okay, then let's talk about osmosis, right? So first of all, osmosis, the concept of osmosis is the same as diffusion. You have low concentration, you have high concentration, and the molecules are moving from high concentration to low concentration. But in osmosis, a couple of things become really specific, right? First of all, osmosis is specific to the movement of water. Diffusion could be the movement of any particle, any molecule, any ion, anything at all. Osmosis is specific to the movement of water. So osmosis is the movement of free water molecules from a region of high concentration to a region of low concentration. So first thing that is changing, or the first thing that is a bit different from diffusion, is that osmosis is going to be specific to water molecules. Another thing that is different between osmosis and diffusion is that diffusion is just taking place randomly. For osmosis, you need to have a partially permeable membrane. If you're just taking two glasses of water, you're taking a juice and a water, and you're mixing the two of them up, that is diffusion. But when you put a partially permeable membrane in the center, that is going to become osmosis. So osmosis is the movement of free water molecules from a high concentration to low concentration to a partially permeable membrane. That is what osmosis is. And when we are talking about osmosis, we can observe osmosis in both animal cells and we can observe it in plant cells as well. Now, before I go into those small little things, right, I want to discuss this particular thing with you, and this is the important thing. Animal cell in osmosis in animal cell and osmosis in plant cell. Let's go ahead and study what happens to plant cells and what happens to animal cells in relation to osmosis. So we have, let's start with our animal cell. Okay, in our animal cell, we are going to go ahead and we are going to take our a red blood cell. Okay, now you are doing this experiment with me in the laboratory. We are going to prepare three types of solution. We will prepare a dilute solution, which is going to have water in it. For example, I told you guys, go ahead, take a glass of water, and this will be our dilute solution. Then we are going to have a normal solution. So what you and I have done is that we went on Google and we checked what is the concentration of sodium chloride in the red blood cell. So we got the answer, for example, 0.9%, that, oh, this is the concentration of sodium chloride in the RBC. So we went ahead and we made that exact same solution with the same amount of sodium chloride. That is the normal solution. It has the same concentration of sodium chloride as the inside of the red blood cell. So inside of the red blood cell, whatever is the normal concentration of sodium chloride, you made a solution of that. And then you made a concentrated solution in which you added a lot of sodium chloride. So that is what you did. One solution is completely dilute, it is pure water. One is the normal solution that has the same amount of sodium chloride as a normal cell. And then you have a concentrated solution. The dilute solution, the solution that is pure water, or a dilute solution, is known as a hypotonic solution. Hypo meaning less, tonic meaning concentration. So it is a dilute solution, we call it a hypotonic solution. So a hypotonic solution is your pure water. Then you have the normal solution, the concentration of the solution is same as the concentration of the red blood cell. This is a normal solution, and a normal solution is known as isotonic. Iso meaning same and tonic meaning concentration. Isotonic solution. And then you have the hypertonic solution where you have added a lot of sodium chloride, a lot of salt. That is a concentrated solution, or we can call it a hypertonic solution. Now, what happens when we are actually doing the experiment? We will start with the hypertonic solution, you and I. Now, you took your red blood cells and you added it in the glass of water. So you said, this is hypotonic, miss, this is the dilute one, and this is complete water, and I'm just going to add red blood cells to it. What are we going to observe? Now, what is going to happen? This is your red blood cell. This is the sodium chloride that is on the inside. Outside, you don't have any sodium, you only have water. So what will happen? Inside the cell, the concentration of water is less. Outside the cell, the concentration of water is more. What is going to happen? The water is going to move into the cell by osmosis. Remember, the cell membrane is a partially permeable membrane. So the water movement is going to be called osmosis. So the water is going to move into the cell by the process of osmosis. What will that do? The cell will start to swell up, because now it is getting a lot of water in it. It is absorbing a lot of water. And because it is absorbing so much water, what is going to happen to it? It is literally going to start to swell up, and it is going to increase in size. And it's going to keep on increasing in size until it will literally rupture. It's literally going to break apart. And the reason it's going to break apart is going to be because it literally swelled up. It swelled up with so much water, and the cell membrane could only take the stress, or it could only take the pressure of water up to a certain point. The water pressure became too much, the cell swelled up, and it burst. It burst, and this bursting of the red blood cell is known as hemolysis. Hemo means blood, lysis means breakdown. Okay, so hemolysis is breakdown of red blood cell. So your red blood cells are being broken down, or they are bursting because they are taking in all the water from the surroundings. This is what happens when you take a red blood cell, or you take a cell from the animal and you put it in a hypotonic solution. Now, what about a normal solution or an isotonic solution? In a normal solution, nothing will happen. Why? Because the amount of water on the outside and the amount of water on the inside is the same. You can use the term water potential, right? Or you can use the term concentration. Concentration of water on the inside and outside of the cell is the same. Same amount of sodium chloride, same amount of water. There is no difference at all. So there is no concentration gradient. So the water doesn't have to go anywhere at all. So it stays where it is. And as a result, the red blood cell will stay completely normal. You won't see anything at all. Last experiment, you took your red blood cell and you went ahead and you put it in a concentrated solution, or the hypertonic solution. Now, in the hypertonic solution, what is going to happen? This is your cell, okay? And this is the situation outside of it. In your cell, you only have a little bit of sodium and the rest of it is water. Outside the cell, you have a lot of sodium and very little water. So inside the cell, you have a high water concentration. Outside the cell, you have low water concentration. Outside the cell, you have a lot of sodium, but not enough water. Inside the cell, you have little sodium, but a lot of water. So what is going to happen? The water will move from low concentration. So the water will move from high concentration to low concentration. Who has the high concentration? Cell. Who has the low concentration? Solution. So the water will start to move out from the cell to the outside. And when the water is going to move out from the cell, what will happen? The opposite. Whatever was happening in the opposite, in the dilute solution, it will be opposite to it. So in the dilute solution, the cell was swelling up. Over here, since the water is moving out of the cell is going to shrink. This process is known as crenation. Crenation is when the red blood cell is shrinking. The red blood cell is going to shrink, and eventually it is going to break away. It's going to end up getting destroyed. So this is what happens to the animal cell in a dilute solution, in a normal solution, and in a concentrated solution. And the reason you need to know this is because you get questions related to it quite a lot. In the topic of movement in and out of cell, this is where they like to trick you up. So they will give you experimental questions, practical life questions related to this particular topic. Any confusion? Does anybody want to ask anything? Monami, Arisha, any questions?
No, miss.
Are you sure? All good? Okay, so this is basically what happens to the animal cell. Now, what is going to happen when we do the same exact experiment, but we end up doing it in the um, to end up doing it to a plant cell? We are going to get surprisingly different results. Just give me 1 minute. Sorry, guys. I have quick. Okay. All right. So when we are going to talk about our plant cell, and we're going to talk about the plant cell once again in your we're once again going to talk about it in relation to all three solutions of yours. Okay. So when we are going to talk about it, we are once again going to have three of the solutions that we have. We are going to have our we have the hypotonic solution, which is dilute. We have a hypertonic solution that is concentrated, and we have an isotonic solution that is normal. So let's take a plant cell and put it in a hypotonic solution. Now, this is your plant cell, and you added it in a glass of water. What is going to happen? The concentration of water is high on the outside, and the concentration of water is low on the inside. What will happen? The water is going to move into the cell by the process of osmosis. Now, water is moving in by osmosis. What will happen? The cell is going to swell up, just like the red blood cell. You can see we are doing exactly the same. The cell is going to swell up, but the plant cell will not burst. Does anybody here remember why? I mentioned something about it. Does anybody know why the plant cell is not going to burst?
The cell wall.
Because
Exactly. Good job. The cell wall is present. The cell wall gives a lot of support to the cell. Because it gives a lot of support to the cell is going to swell up, but the cell wall will not let it burst. It will not let it break apart. And instead, what will happen? The cell will simply swell up and it will end up becoming firm. It will end up becoming hard and firm. Right? So that is what you're going to see. You're going to see that the plant cell is going to swell up and it's going to end up becoming firm. There is a special term we use for it, and that term is turgidity. Turgidity, or we can say that the cell has become turgid, depending upon what what the statement is. So we can say the cell now has turgidity, or the cell has become turgid. And why has the cell become turgid? Because of the pressure of water inside the cell. The cell wall is not letting it burst, and the pressure of the water inside the cell is basically making it swell up, and when it becomes swell up, it becomes firm, and that we say that the cell has become turgid. And the pressure that is inside the cell, the pressure that is inside the plant cell is known as turgor pressure. So same thing, we're using the same term, turgidity or turgid, but simply it's known as turgor pressure. So turgor pressure will develop inside the cell. It will make the cell swell up and become firm. The cell will not break because of the cell may fall, and as a result, the cell will become firm and it will become turgid. The term we use is turgor pressure or turgid or turgidity, depending upon what kind of English you're writing or what kind of statement you're writing at that time. And when we are talking about it, where is all of this water going, by the way? What part of this plant cell do you think this water is going?
The vacuole.
The vacuole. Because remember, the vacuole is responsible for maintaining the osmotic pressure, for controlling the osmotic pressure, for controlling the water pressure. That is the job of the plant cell, the vacuole, sorry. So that is where the water goes. It goes into the vacuole. The plant becomes turgid, and that is what will happen. Now, isotonic solution, what do you guys think will happen in isotonic solution?
The thing will stay the same.
It will say the same, exactly. It's a normal solution, which means the concentration in and out of the cell is the same. So if concentration in and out of the cell is the same, it is going to remain as it is, and as a result, you are going to have you are going to see nothing is going to change. It's going to be completely normal. Then what happens when you put the plant cell in a concentrated solution or in a hypertonic solution? Okay, this is your plant cell and you have put it in a concentrated solution. What is going to happen? The concentration of water or the potential of water is going to be higher inside the cell and it will be lower outside the cell because the solution is concentrated, so it has a lot of solute and not enough water. So what is going to happen? The concentration of water or the water potential is higher inside the cell and it is lower outside the cell. Water will start to move out of the plant cell by the process of osmosis and the plant cell will shrink. Same story as red blood cell, but again, when you reach the shrinking part, story changes. It will not shrink in the same way as the red blood cell. It will shrink in a completely different way. It will shrink like this. The cell wall stays exactly where it is. The cell wall is not moving. Cell wall is staying exactly where it is. Who is moving? The cell membrane. The cell membrane is going to start to pull away. It's going to start to detach. It's going to start to pull away from the cell wall and it's going to slowly start to shrink inside. It is slowly going to start to move inside. That is what is going to happen. If you look at it, you will see the size of the vacuole is also decreasing. The size of the vacuole is decreasing. The cell wall is going to pull away, sorry, the cell membrane is going to pull away from the cell wall and it's going to start to shrink. So the cytoplasm is shrinking, the vacuole is shrinking, the cell membrane is shrinking, but the cell wall will stay exactly where it is. This is where it is different than red blood cell. And this particular thing that you're happening, this process, what is this process called? We will say the plant cell has been plasmolysed. This is the term we used for it. The plant cell has been plasmolysed. The plant cell has undergone plasmolysis. This is what it is called, plasmolyze. And what will actually happen to the cell? The cell will become flaccid. When we were putting it in water, it was becoming turgid and stiff and firm. When you put it in a concentrated solution, it will shrink, it will become flaccid and soft. That is what you're going to see. You take a slice of potato and you will see questions like this in past papers, and you put it in a concentrated solution, you take it back out, it's going to be floppy. It's going to be all floppy and soft. The reason it's going to be floppy and soft is because all of the water has been removed from it. If you do any kind of cooking, or if you've ever done a cooking, or if you guys want to do the experiment later on, take a piece of, for example, cucumber, put a lot of salt around it and leave it. Come back after like half an hour and you will see that the cucumber has become really, really soft. Why has it become soft? Because now there is salt all around it. The water will move out, it will end up becoming flaccid, it will end up becoming plasmolized, and it will be soft. So this is what happens to plant cell in relation to osmosis. Any confusion? Does anybody want to ask something?
Miss, so flaccid
Yeah.
is the opposite of turgid, right?
Exactly.
Okay.
Turgid will be hard and firm. This is going to be extremely soft and flaccid. Okay, all right. So now that we know what is going to happen to the plant cell, and that is what you have to know about your topic of osmosis. Now, another thing that you have to know about, the final thing that you have to go over in the topic is active transport. Okay, what is active transport? So up till now, we were all talking about the fact that you are moving from high concentration to low concentration. That is all we were talking about, high to low, high to low. Now, you could ask me, how can we do the opposite? What if I wanted to go from low to high? That happens in the body as well, and that is known as active transport. Both osmosis and diffusion are passive processes. Passive meaning you don't need energy. Active means you need energy. And to explain this, just imagine you're climbing up a hill or you're climbing up a mountain. You need a lot of energy to climb up mountain, right? I mean, climbing down is very easy, but when you come to climbing up, it's really hard. Why? Because you're going against the gradient. Because you're going against gravity, you're going against the gradient. So the same logic applies over here. You have an area of high concentration, you have an area of low concentration. So you're moving up the particle from low concentration to high concentration. And to do that, you need to have energy. So active transport is an energy dependent process. Your osmosis and diffusion are passive. That means even if you don't have any active energy, it will still work. But for this, you need to have energy present. And what is active transport? How will you guys define it? Active transport is the movement of particles from low concentration to high concentration against the concentration gradient. When I told you diffusion, I said down the gradient. Down the gradient means that you're going down. Now we're going against, we're going in the opposite direction. So against the concentration gradient, using energy. Using energy is also important because if we don't have energy, active transport will not work. So we need to have special energy for it. So when you are talking about it, there are in our body, we need active transport. We need to be able to take it from low into high. Let me give you a big example of active transport in our body, glucose. Glucose moves into our cells by active transport. So imagine that there is already a lot of glucose in the cell. Let's imagine you're running, okay? So you're working out, you're exercising, your cells want more and more and more and more glucose. Now imagine that there's already glucose inside the cell, and so if you did not have active transport, the glucose would simply diffuse out. The glucose would simply move out, right? But that is not going to work for us because we are running. We need that high amount of glucose. That is why we need active transport. That even though there's already a lot of glucose inside the cell, but we need to keep bringing more and more and more in so that the cell can break it down and give you energy so you can actually run. That is certain instances where active transport is necessary. A lot of processes in your body, for example, your nervous system. In your nervous system, when you're talking about your nervous system is where the electrical impulses are being conducted. A very important ion for the conduction of electrical impulses is sodium. So what happens is that we have this process of active transport that keeps on pulling the sodium inside the nerve cells. Even if the nerve cells have a lot of sodium, it will keep on pulling all the sodium inside the cell so that the nerves can keep on functioning properly. So this entire process is basically known as going to be your active transport. And how does active transport actually work? So you have this special molecule. It is known as a transport protein. Okay. You have a special molecule, a special transport protein. Think of it like a gate. Okay. And you have this gate, and this gate will be present in your cell membrane. So in your cell membrane, we have special proteins. They are known as transport proteins. Now, what is going to happen? This is, for example, your glucose. Your glucose will come and bind over here. It will come and bind to or will come and attach to the transport protein. Now, what will happen? This transport protein will use energy. It will take up the energy. If you remember, I told you that the energy in our body is in the form of ATP. When I told you guys about mitochondria, I told you the molecule for energy in our body is known as ATP. So that ATP will come, it will be used by the transport protein. And what will the transport protein do? It will turn upside down, or by using the energy, it will flip upside down. It will change its shape. It can do it in different ways, but what will it will basically do is that it will change its shape. By using the energy, it will change its shape, or it will turn upside down, or it will do something, and it will move that glucose to the inside of the cell, and the glucose can then move away. This process is going to be known as your active transport, right? So you have your substance, you have your molecule, you have a transport protein. The molecule or the particle will attach to the transport protein. The transport protein will use energy. It will change its shape, or it will turn upside down, or whatever, and it will move that particle to the inside of the cell. Once the particle is on the inside of the cell, it can simply then go into the cytoplasm and do whatever it needs to do. Any confusion?
No, miss.
Okay. So, okay, yeah, another example of, by the way, this active transport is in your plant cells. So if you look at the root cell of a plant, or if you look at the root hair cell of a plant, so water moves into the plant cell by osmosis, but the mineral ions are taken in by active transport. Let's be clear about that, because you get questions on that. Water will move into the root by osmosis, but mineral ions will move into the root by the process of active transport. So that is basically it for the topic of movement in and out of cell. That is all we had to cover. Now I'm going to go ahead and share a few questions with you guys, and we're going to go ahead and we're going to discuss them. Just give me a second. Excuse me. Okay, go ahead, take a look at the first question. Give it a read and let me know what do you think the answer is going to be.
It's the movement of water molecules from a region of high water potential to low water potential down the
Yeah,
concentration gradient.
yeah, and you have to add one more phrase to it.
Through a partially permeable membrane.
Exactly. You have to say through a partially, that's why it's a 3-mark question. Even though it's a definition, they have given you 3 marks for it. 1 mark from high water potential to low water potential. 2nd mark down the potential gradient. 3rd mark through a partially permeable membrane. So you need to say all 3 things to get all 3 marks. Okay, go ahead, give this question a read and let me know when you guys are done.
Miss, can you scroll down?
Yeah, sure. Monavi, are you also done reading?
Yes, miss.
Okay. Question states, predict which of the liquids would cause the largest decrease in the mass of the potato. What do you think? Who would cause the largest decrease in the mass of the potato? And also explain why. Any idea? Any idea at all? Whatever, you don't have to be correct. Like whatever you think it
The
should be.
1.01.
Yeah, good job. Can you explain why?
Because the sodium concentration outside is more than the
Yes,
sodium concentration inside, so the water
exactly.
leaves the cell.
Exactly, good job. So 1 mole per dm cube solution is the most concentrated, right? So the water potential outside the cell will be low. The water potential inside the cell is high. So the water will move from the inside of the cell to the outside of the cell. That is why the potato is losing mass, because the mass that it is losing is actually the water. It's losing water mass. So the water is moving out of the cell. That is why the potato stick is losing mass, because it's losing water. So in the 1 mole per dm cube solution, it will lose the most amount of water. So you will see the greatest change in mass over there. The student dried the potato sticks with paper towels before putting them on the electronic balance. Suggest why. What do you guys think? Why did he dry them and then measured their weight? Why not just measure them as it is? Any idea? It's just a 1 mark question, because this is a pretty general
Because
sort of a common sense question. There's no theory behind it. Yeah.
water is like an insulator with electricity, so it wouldn't react well.
An electronic balance simply means that it's going to be using electricity to work. It should not, having water there should not. Okay. First of all, water is not an insulator. Water is a conductor of electricity. And secondly, when you are talking about it, um, water is water is not going to have an effect on the fact that it's an electronic balance. An electronic balance simply means that it's using electricity to work. That's it. So the reason that they said that the student did that is to remove excess water from the potato. By removing excess water from the potato, you make sure you get an accurate measurement of the mass. If you place a wet potato on the measuring balance, you will also be including the mass of excess water that is stuck to it. So that is why you were drawing it. It's not a theory-based question. It's more of like a common sense question or a general question, that why would you dry it? Because you want to remove all the excess water to make sure that your measurement is as accurate as possible. Okay. Take a look at this one. Take a look at part C. Give it a read and let me know what you guys think the answer should be.
Since water moves out of the cell,
Yeah,
the cell membrane starts to shrink with the vacuole and the chloroplast, and the cell wall
exactly.
stays the same. Uh,
Yeah,
this causes the cell to become flaccid, which causes the cell to be floppy and soft.
exactly. Good job. Okay, just a couple of things. So your answer is correct. You don't need to say it like the cell wall stays where it is. That the way you should word it is that the vacuole is going to shrink. the cell wall is going to get detached from the cell wall and it's going to shrink as well. And that causes the cell to become plasmolized, and that is why it is becoming soft and flaccid. So plasmolized is important, flaccid is important. These two statements you have to mention because they'll get you the actual marks. The student followed the same experimental procedure with boiled potato sticks and found no overall change in the mass of the potato. Suggest why the mass of the boiled potato sticks remained the same. What do you guys think? What is happening when we're boiling it?
Water goes in a potato.
They're saying they're doing the same experiment. That means that they're first boiling the potato sticks and they're taking the same boiled potato sticks and they're putting them in the same solution. And they're not getting any result at all in any of them. The reason this is happening is when you're boiling the potato sticks, you're damaging the cell structure, right? If you're going to boil the potato sticks, the plant cell is going to get damaged. If the plant cell is damaged, are you going to see any osmosis? No, there will be no cell wall, no cell membrane. Everything is going to be broken down because you boiled it. So by boiling it, you're actually destroying the plant cell. That is why first they did the experiment using raw potato. At first, they did not use cooked potato, they used raw potato. But when they boiled it, when they cooked it, you destroyed the plant structure. There is no cell wall, no cell membrane. If there is no plant structure, then what is going to happen? You put it in any solution, put it in water, put it in salt, put it in whatever you want. You won't see anything because the plant structure is not there anymore. Making sense?
Yes, miss.
Munavi, making sense?
Yes, miss.
Okay. Hold on 1 second. Okay, take a look at this particular question, give it a read, and let me know what you think the answer is going to be.
Diffusion moves from an area of high concentration to an area of low concentration.
Good job. Good job.
And then diffusion is how uses passive energy, whereas active transport
Yes.
is active.
Yes. Don't say passive energy. There's no such thing as passive energy. Just say it's a passive process or it's a passive process. Yeah, it's a passive process. Passive automatically means no energy. Passive is not a type of energy. If you say passive energy, it's like you're saying one type of energy is passive. No, it's just passive means no energy. So it's a passive process while active transport is going to require energy. Okay. Explain how roots are adapted to absorb ions. What do you guys think? We discussed this in our class when we talked about the cell. I told you guys how it is adapted.
They have large surface area.
Good job. So they have that long root hair cell has a long hair-like projection that is extending into the soil or that is going between the soil particles. That gives it a large surface area. And what did we just study today? Larger the surface area, faster the rate of diffusion. So faster the rate of diffusion, the more water will be absorbed in. And also it has the transport proteins to allow active transport so the mineral ions can be absorbed. Okay. Yeah, I think this is a longer question. Okay, guys, so I think we're going to go ahead and just stop it over here for today. So we have covered the theory portion related to related to our movement in and